go back to the days that I had you. Back to one more night like we used to. I miss every minute we've been through. From the hardest part to the best you. Make the stars align. Words cannot describe. Don't need no more goodbyes. You leave me hypnotized. Make the stars align. I need you here tonight. Don't need no more goodbyes.
So, like I said earlier, the phylogeny is the evolutionary history. So, how do you correctly build this phylogeny information? Traditionally, it has been dependent upon many the comparative study using the appearance. So, morphology is how it looks like. Anatomy, internal structure. So based primarily upon this morphology and anatomy comparison, and occasionally, whenever it's possible, with the help of the fossil record, to have this complete story about this phylogeny, was the way they have this the study of phylogeny correctly. So, for example, if you
The 65,000 species of vertebrates found today on land and sea all derive from a common ancestor. We can see the evolutionary relationships between all of these organisms in a phylogenetic tree of vertebrate evolution from the last common ancestor, which was probably a jawless fish that lived about 530 million years ago. In the first great branch of the phylogenetic tree of vertebrates, jawless fish evolved into fish with jaws. Forever after, the jawless fish and the jawed fish would evolve separately. The jawless fish are still with us in a small group of about 120 species that includes lamprey eels. In the next great branch for jawed fish, bony fish split from fish that have cartilage skeletons instead of bone, such as the sharks, rays, and skates that are still with us. The bony fish evolved bodily structures such as lobed fins, which evolved into the modern limbs of amphibians, reptiles, and mammals. Each branch in the phylogenetic tree represents a common ancestor for the lineages that rise above the split. Above the most recent branch that we show, when reptiles split from mammals, there are 5,500 known species of mammals and 10,000 known species of reptiles.
So, bottom line is that there are so many variety of the different species. Then, how were you able to actually correctly interconnect according to the evolutionary relationship, connecting correctly each of which the common ancestor was? Sometimes actually you have some of those clues of, for example, as you can see in this particular form of salamander, still living salamander, having four limbs, salamander as a salamander, but also fish-like, and even more strikingly, the gills. Instead of the lung, this salamander have these gills. Through these gills, they actually have this gas exchange. They breathe. Axolotl. I don't know why this axolotl has the Korean name. What is this? Holotl. I don't know. But anyway, sometimes that is lucky. You get luckier than the other days, but not every time actually you can have this an easier lucky break. So such the comparison, and one of the major tools by which you can make a correct comparative study to assign any specific evolutionary relationship is to use the homologous structure. The homologous structure is a structure or the shape which reflect the same evolutionary origin. So when you look at this bat, a wing, but if you look closely, the anatomy and the morphology of this bat's wing have something actually, so we can get some clue about its evolutionary origin. We will just get to that it later today, in a minute. So the point is, among different organisms has sharing the homologous structure. If some of the organisms share the homologous structure, that means they are highly related or more closely related evolutionarily, but that does not necessarily mean that those homologous structure look exactly the same. They could look different. And this is something that indicative of the divergent evolution in the form of this adaptive radiation or anything like so. Adaptive radiation is another a good example of such divergent evolution. Starting from common ancestor, you become differentiated, and you will see in most cases of this homologous structure, you will see such diverging features. So here is the one famous example of such a homologous structure in the forearm of a human and the leg of a cat and this fin. I usually used to call this fin, but I shouldn't have. There is actually more correct name of calling this the fin-like structure of this whale. I think there's a flipper or anything. I learned from the today's film that I'm going to show you. And a bat's wing, they are all homologous structure, even though, especially in the case of these whales and bat's wing, they are totally look different. But the point is, if you look closely inside the anatomical feature of these bones, they are all actually like having the same origin. As you can see in the different type of bones are colored in the same colors. So that's the homologous structure. On the other hand, the analogous structure is they look similar, the shape, and more importantly, is the same function. But they are the one actually found in unrelated. Evolutionarily, they are unrelated among these organisms, but the very similar looking structures are found, and more importantly, their structures are the reason why they look similar is they are playing the same function. Remember, the shapes dictate the function, right? So that's the two contrasting examples between homologous versus analogous structure. So this, on the other hand, in the case of a homologous structure which reflect the so-called divergent evolution, this analogous structure reflect the convergent evolution, starting from two different origin to converge on into the same similarly looking thing is what this convergent evolution means. So here, four different type of this wings of the insect, wings of the dinosaurs, some of the flying dinosaurs as a pterosaur, pterosaur, the pterosaur, the pronunciation, and this bird feather, the wings, and bat's wing, which are all different actually, but they all look alike, and then they provide all the same identical function. Is a perfect example of such analogous structure. So let's take a look at about more clear explanation about such things here.
In these pictures, we might look at the fin of the shark and say they are very similar to the flippers of a dolphin, For the dolphins, we don't call them as fins, we call them as flippers. They look very similar and they're both used for swimming, so we could say, hey, maybe they are very related to each other. And similarly, if you look at the wings of the bat, let me use a different color, wings of a bat and the wings of, say, an eagle, again, both are used for flying, they look kind of similar, we might say they are related to each other. But it turns out that in reality, they are not as closely related to each other. And in fact, it turns out, surprisingly, it's these two structures are more closely related than these two or these two. Yeah, this sounds very confusing, I know. And so in this video, we'll explore why certain structures which might look similar and perform same function did not necessarily mean that they are related to each other. And on the other hand, we will see that although some other structures might look very different and perform different functions, they can be related to each other. To avoid confusions, let's get rid of this picture and start fresh. So if you take a look at the wings of a bat, the flippers of a dolphin, and our hands, then although they look very different and they perform different functions, you look carefully, look at their bone structures, you'll find the bone structures are very similar. Let me show you. If we zoom in a little bit, let me zoom in to the wings and the flippers and the hand. And here are the bones. Look at them. Approximately, this is how the bones would be. Look at them. Do you see similarities? To make it more clear, let me color them appropriately. So if I color them, now look at these bones carefully. Over here we see one bone, one bone, one bone. And this now is an elbow, our elbow. You get two bones, two bones, two bones. Then we get the wrist bones, wrist bones, wrist bones. Then you get the hand bones over here, 5 of them. There are only 4 here. Again, 5 of them. And then we have fingers, finger bones, and finger bones. So can you see how similar structures they are? The number of bones and the kind of bones are very, very similar to each other. Of course, yeah, there are, of course, some differences. There are different species altogether, so some differences are fine. But because of so much similarity, we can say that all these bones evolved from a common ancestor because of such similarity in the structure, right? So let me just write that down. Let me zoom out a little bit. Same thing, I zoomed out a little bit. And now we can say that all these bones, they have evolved from a common ancestor. So same structure from the common ancestor performing different functions. Okay? Now let me give you a separate example, an extreme example in plants of the same thing. If you were to look at these 3 plants, this is a pitcher plant which contains the pitcher, and what it does is if insects fall into that, it will close the lid, it will not allow it to escape, it will start digesting it. And this is a Venus flytrap. It's like a mouth-like thing, so if a fly comes in, it basically closes the mouth and starts digesting it. And this is our cactus, which has a lot of spines, or the thorn-like structures, they're called spines, pointy spines. Now what's interesting to see over here is that if you do careful research, if you do a careful study on these structures, then we can see that the picture of the pitcher plant, the mouth of the Venus flytrap, and the spines, they are all actually highly modified leaves. Now I know it's hard to believe how a leaf can modify into these different things, but it turns out to be true. We'll trust our biologists, okay, on this. So again, what does this mean? Since they are all modified from the leaf itself, from the same leaf itself, we can again say, hey, that means these structures also must have come from a common ancestor. Common ancestor. So with these couple of examples of evolution, what is the common underlying feature you can find out in these examples? What is common in these evolutions? Can you think about it? Well, in both these examples, we see structures are evolved from the same ancestors, but they end up performing different functions. See over here, They perform flying and swimming, and our hands can do so many things like I don't know, maybe scratching or climbing. Over here, these are used for catching insects. Here also they catch insects, but the spines are used as a protection. So different functions, but same ancestry. Such structures, which have different functions, but same ancestry, we call them homologous structures or homologous function. I'll just write homologous, okay? So what's important is that the word homo means same. Same what? Same ancestors, and that's important. Same ancestor, but they can have different functions. Can have different functions. And I say the word they can have different functions is because they can also have same functions. For example, if you compare our hands with the hands of a chimpanzee, okay, they're also homologous. I have not drawn the bone structure, but bone structures will be very similar, and chimpanzees' hands also perform the same function as our hands. So homologous structures can perform same functions. They can also perform different functions, as we see over here. What's important though is that they have the same ancestry. Same ancestry. On the other hand, if you compare the wings of a bat and an eagle, let's see what we get. We've already seen the wings of the bat. We already saw the bone structure of this. Now let's look at the wing of an eagle. If I zoom in and I look at the bone structure, it looks somewhat like this. So again, we see one bone here, then we have two bones over here. You can see the wrist and a little bit of that as well. And when it comes to fingers and hand, that's where things get a little different. But again, the bone structures are pretty similar. So we can say the four limbs means, the front limbs. In humans, four limbs are just hands. So the four limbs, we can say, still came from the same ancestry because of the same structure. So we can still say the four limbs of the bat and the four limbs of the eagles, they are homologous. Okay, the structures are the same. But let's look at the wings now. That's important. Concentrate on the wing structures. For the bat, look at how the wings are formed. The wings are actually skin. This is the skin of the bat. And the skin of the bat kind of makes a web over here, right? It's attached to the fingertips. Can you see that? The skin is stretched over the fingertips, and that's how they have evolved, because of which they can fly. But look at the wings of the eagle. These are not skin, these are feathers. And the feathers are attached to the entire arm. They're not web-shaped like in the case of the bats. They're not skin. They're not connected to the fingers like in the bats. So a completely different structure if you look at the wings, isn't it? So can we say that these wings might have evolved from the same ancestor? I don't think so. Because they have such different structures, we can now say that maybe this wing evolved separately from some ancestor. I'm going to call this as ancestor 1. May have evolved from some ancestor, and this wing might have independently evolved from a different ancestor altogether. And so are the bat wings and eagle wings homologous? The answer is no. Because they have such different structures, they must have evolved from different ancestors. So they are not homologous. So if they are not homologous, what do we call them? Now, before I tell you the name of this, let me show you another example. If you look now at the flipper of a dolphin and compare that with the fin of a shark, then if you look at the bones of the shark, it might look somewhat like this. This is an approximate drawing, okay? And if you're wondering why I haven't colored it, mainly because there's nothing similar between them to color. Okay, these are made of bones, very similar to our hands. These are not even bones. These turn out to be made of cartilages. Very different structure. So because they have such different structure, we can now again say, hey, they must have evolved separately from different ancestors. So this must have evolved from a different ancestor compared to this one. These are separate examples, okay, not related to each other. So now again, if you look at these two, what common feature do you find in this kind of evolution? In this, we see that structures come from different ancestry, but they perform the same functions. Such structures are called analogous structures. Analogous structures or analogous organs. Okay? What's important over here, they have different ancestors. Different ancestors. Different ancestors. But the important thing is that they have the same functions. They carry out same functions. Have to carry out the same functions. Like over here, both of them swim. Over here, both of them are used in flying, but underlying structures are super different, therefore different ancestors. So what is the big picture that I'm trying to paint over here? Well, the main thing that I'm trying to tell over here is when we are studying evolutionary biology, just because we see certain structures which look very different at first and perform different functions, they can still have the underlying same structure, and so they could still be related by a common ancestor, homologous structures. And on the other hand, just because we find some structures in some species to be very similar, it look very similar and they perform same function, need not necessarily mean that they are related to each other. It's totally possible that they evolved independently from separate ancestors altogether. And so these are called analogous structures. Now, one last thing before we wind up is that if you look at the homologous structures, you see it from one common ancestry, from the same underlying structure, we are getting different functions. So diversification is happening over here, right? Same structure is used for many different things. So homologous structures give us something called divergent evolution. All right, same structure used for many different things, many different functions. Divergent evolution. On the other hand, if you look at analogous structures, they're exact opposite. Two completely different structures from completely different ancestry have the same functions, have evolved independently to give us the same functions. So different structures are coming together sort of to give us the same function. So it's like converging. This evolution is converging, right? Converging to give same function. So we call this convergent evolution. So analogous structures are evidence for convergent evolution. All right? Convergent evolution. That's pretty much it.
Okay, that's pretty much of it. It's a very clear and detailed explanation about these two differences. So, those are some kind of a comparative morphology and anatomy. But also, on top of that, a lot of those fossil records have been such a valuable asset, instrumental in actually clearly, correctly deciphering the common ancestors. So, like here is all the fossils of echinoderms, and several hundred million years old, this dragonfly, basically not showing any drastic big major changes. And also some of the ancestral form of the reptile fossil, fish. Also, as you can see, the overall body plan of the several hundred million years, it has just a preserved body pattern. Sometimes, not only those that stand out, the fossilized form, sometimes it's a captured amber fossilized insect, sometimes it's a trapped, which is actually because many occasions multiples of such insect as well as their clues about their habitats are also captured in this sap. Amber is the fossilized the plant sap, and so if you are lucky enough to obtain this, then you can have actually a lot of the information out of this. Sometimes these dinosaurs' eggs also were recovered in the form of the fossils. Okay, so many such fossils, all of which are the reason why they are so valuable is they can provide this common ancestral form, much more details form, so that it becomes much more easier to connect or those are seemingly unrelated organisms living present too. So those are the value of this fossil record. One of such valuable fossil record is Archaeopteryx. In actually specific Latin way of pronunciation is an Archaeopteryx, but the English influence usually people pronounce this as something Archaeopteryx, meaning simply ancient feather or wings. And it's been for a long time. It's been recognized as an intermediate form of the reptiles and birds. So, like, been identified as an ancestral form of the bird evolution. What am I talking? Bird evolution. But then, several years ago, like just the it's been heavily criticized by some of the due to some of the very trivial factors, and then it's been heavily exaggerated so that in this country it's been like temporarily, although it is a temporary basis, that or the description of this Archaeopteryx in the high school textbook, biology textbook, has been all eliminated by and what actually those who were involved, like scientists, Korean scientists, and those who can make difference, were not even aware of that instance. But then only after this was reported in this renowned scientific journal Nature, then only then those scientists were aware of it and then fought back to reinstate the description. But anyway, the point is with some of the unconfirmed, a little bit of the detailed miscellaneous things that we need to actually further make sure. But obviously, this Archaeopteryx could be regarded as such a the intermediate form between the transition from reptile to birds. However, the way scientists present this information in the biology was somehow a little bit, maybe it was a little bit of mistaken, misleading, and so gave a little bit of the opened the way for such invited attack and criticism. That's how it happens. But anyway, so in other words, it can be these Archaeopteryx could be placed somewhere near in this position, which in many features actually support such notion. On the other hand, pterosaur, the actually flying the dinosaur, have a long further divergent origin, so that if we keep following their common ancestral origin, common ancestor, then you have to go back further to the past. So in other words, the Archaeopteryx and other form of flying dinosaurs are way distantly related. Okay. Another very interesting such a fossil was Tiktaalik or Tiktaal. It's an intermediate form between fishes and amphibians, so that four-legged fish, you can describe this. And this one actually has such a preserved the bony anatomy with all those land mammals and reptiles. So obviously it's an indicative of such an common ancestor of all those land four-legged animals like that. So this can be viewed as such a so-called like missing link between this is about the whale. The one famous feature about these whales and dolphins was that they are mammals, and they are the one who actually went back to, once they have the after living the land animals as a land animal, somehow they have actually evolved into the marine form of this living features, so that they became like fish-like organisms, and those are also been kind of recorded in the form of fossils, so that from top to bottom in actually similar location, which is actually in Pakistan. So like highly similar form of some of the like transition from like the fossil was identified. Like if you look at this internal anatomy of this, like compare and also compare with that of whale, present day whale, and that fossil, you can see some of this transition clearly. And further in the deep down of this fossil record in geology time scale, you could also see also like a time according to the timeline, respective timeline. These two other forms, more ancient form of those ancestral form of those whales, were also found in Pakistan too. So if you follow this bone anatomy of these two fossil forms, then you could be fairly convinced that those were the way actually how the transition and evolution has occurred to finally become the whale, present day's whale. So let's take a look at this one.
안녕하세요, 북툰입니다. 저는 지난 영상에서 중간 단계 화석이 없다는 주장은 창조과학 측의 가짜 뉴스입니다 라고 말씀드렸습니다. 그 뒤로 많은 분들이 그러면 중간 단계 화석의 예를 보여달라는 댓글을 달아주셨는데요. 저는 몇몇 분에게 답변 대신 책 화석은 말한다를 권해드렸습니다. 왜냐하면 중간 단계 화석은 구글링으로도 얻을 수 있을 만큼 자료가 많지만 이 책이 풍부한 해부학적 설명을 곁들여 중간 단계 화석들을 잘 정리해두었기 때문입니다. 네, 어쨌든 오늘은 중간 단계 화석들을 영상으로 한번 소개해 보겠습니다. 먼저 중간 단계 화석이란 무엇일까요? 쉽게 떠올리자면 중간목, 중간다리같이 진화 과정을 한눈에 보여주는 화석. 물고기에서 양서류로, 양서류에서 파충류로, 파충류에서 포유류로 넘어가는 전이 과정을 입증하는 화석들이 중간 단계 화석에 해당될 겁니다. 사실 중간 단계 화석이란 진화를 직선형 개통도만으로 생각하다 보니 더 크게 부각되는 의미이기도 합니다. 직선 개통도로 보면 생태계를 묶는 방식이 지극히 단순해집니다. 그래서 빠진 고리가 더 부각되고 모든 생물이 정교한 형태로 진화할 것이라는 오해를 낳기도 합니다. 그러나 진화는 높은 단계로 올라가는 사다리가 아니라 수많은 개통들이 서로에게서 자라나 뻗어나가는 나무와 같습니다. 나무로 보면 방향성이 아니라 다양성이 보이고 그 속에서 생물의 전이 과정도 자연스럽게 이해되는 것입니다. 나무 개통도에 대해선 기회가 되면 다른 영상에서 자세히 다루어 보기로 하고, 오늘은 최대한 직선 개통도로 표현해서 중간 단계 화석들을 한번 소개해 보겠습니다. 어쨌든 나무에게도 중간 단계 화석은 필요하니까요. 그럼 워밍업 차원에서 중간다리, 중간목 같은 화석부터 살펴볼까요? 기린이나 브라키오사우루스의 긴 목은 다윈의 자연 선택설을 설명할 때 빠지지 않고 등장하는 단골 예시입니다. 그러다 보니 진화론을 반대하는 쪽에서도 중간목 화석이 없다는 점을 단골로 강조해 왔습니다. 하지만 목이 긴 용각류 공룡의 중간 단계 화석들은 이미 오래전에 발견되었습니다. 기린의 중간 단계 화석도 발견은 오래전에, 연구 발표는 최근에 이루어졌습니다. 목이 길어지는 전이 과정은 진화론으로도 화석 기록으로도 잘 설명되고 있습니다. 어떤 생물들은 화석 발굴 자체가 어려운 종들도 있습니다. 대표적인 예가 뱀입니다. 뱀은 수백 개의 섬세한 척추뼈와 갈비뼈로 이루어져 있고 대개는 부서지거나 분리되기 때문에 완전히 연결된 화석이 발굴되기가 극도로 어려운 생물입니다. 그럼에도 불구하고 4발 달린 도마뱀에서 다리 없는 뱀으로 전이를 보여주는 멋진 중간 단계 화석이 발견되었습니다. 2007년에 발견된 아드리오사우루스는 흔적만 남은 앞다리와 완전한 기능을 하는 뒷다리를 가지고 있었습니다. 2006년에 발표된 나자시 화석은 다음 단계인 흔적도 없는 앞다리와 기능을 하지 못하는 뒷다리를 가지고 있었습니다. 둘 사이를 이어주는 화석들은 그보다 먼저 발견되었습니다. 참고로 힘들게 발굴된 뱀 화석 중 어떤 것에도 발성 기관이 있었다는 흔적은 없었다고 합니다. 뱀과 함께 세트로 창조과학 측의 단골 메뉴에 오르는 생물은 거북입니다. 1992년에 브리타니카 백과사전에는 거북의 중간 단계 화석이 없다는 내용이 있었는데 창조과학 측은 이 내용을 강연 자료에 자주 인용합니다. 하지만 중간 단계 거북은 2008년에 발견되었습니다. 오돈토켈리스는 배딱지는 있고 등딱지가 없는 절반의 거북 화석이며 다른 파충류와 거북 사이의 전이 과정을 보여주는 중간 단계 화석입니다. 브리타니카에는 이 내용이 업데이트되었습니다. 2008년에는 개구리와 도룡뇽의 특징을 모두 가진 중간 단계 화석도 발견되었습니다. 그래서 게로바트라쿠스는 개구롱뇽이라는 애칭으로도 불렸습니다. 개구롱뇽은 몸은 도룡뇽인데 개구리처럼 머리는 짧고 주둥이는 둥글며 큰 눈과 큰 고막이 있습니다. 또 다른 화석 트리아도 바트라쿠스는 현생 개구리의 모습과 조금 더 닮았지만 도룡뇽의 특징도 아직 가지고 있습니다. 원시 양서류에서 현생 개구리의 과도기 과정도 화석으로 잘 채워지고 있습니다. 네, 워밍업이 끝났다 치고 이제 중간 단계의 시조 중의 시조를 한번 알아볼까요? 학명 아르카이옵테릭스 리토그라피카, 일명 시조새. 시조새 화석은 1860년에 처음 발견된 뒤로 현재 12개의 화석이 있습니다. 전 세계 학계에서는 시조새를 대표적인 공룡과 새의 중간 단계 화석으로 인정하고 있습니다. 상징성이 크다 보니 시조새는 집중 공격도 많이 받았습니다. 창조과학 측은 그동안 시조새 화석이 조작이라고 왜곡하거나 중간 단계가 아니라 그냥 새의 한 종류일 뿐이라고 주장해왔습니다. 새라고 주장하는 근거는 외형입니다. 크기가 작고 날개가 달려있다는 이유입니다. 하지만 공룡이라고 모두 크지 않고 날개가 있다고 모두 새가 아니라는 것쯤은 초등학생도 아는 상식입니다. 화석 분류는 결코 외형만 따지지 않습니다. 해부학적 구조를 엄밀히 파악해서 종별 유사성을 분석하고 현대 들어서는 분자생물학의 도움도 받습니다. 시조새는 해부학적으로 새보다 공룡에 더 가깝습니다. 골격은 대부분 공룡과 닮았고 현생 조류에는 없는 이빨과 긴 꼬리뼈가 있습니다. 대부분의 수각류 공룡처럼 손가락이 3개이고 가운데 손가락이 깁니다. 반달형 손목뼈와 중간 발목뼈 관절 같은 공룡, 익룡, 조류의 공통으로 나타나는 특징 외에도 100여 개의 해부학적 특징들을 분석해서 시조새를 수각류 공룡과 고등 조류의 과도기 단계로 분류하는 것입니다. 공룡과 새의 중간 단계 화석이 시조새뿐이라 하더라도 모자람이 없겠지만 지금은 시조새 말고도 중생대 과도기 조류 화석이 수십 가지가 있습니다. 새로 발견되는 과도기 화석들 덕분에 비조류형 수각류 공룡과 고등한 조류 사이의 빈틈 대부분이 채워졌습니다. 새로운 중간 단계 화석들에 대해 미국의 창조과학 측은 대체로 무반응으로 대응하고 한국의 창조과학은 아직도 시조새에만 집착하고 있습니다. 새의 시조를 알아보았으니 이번에는 육상동물의 시조를 한번 알아보겠습니다. 바로 물고기와 4발 달린 육상동물 사이의 중간 단계입니다. 1881년에 발견된 에우스테노프테론은 척추 구조와 머리뼈의 패턴, 지느러미 등에서 4발 동물의 해부학적 특징을 가진 육기 어류였습니다. 에우스테노프테론은 오랫동안 육기 어류가 4발 동물로 진화한 유일한 증거로 알려졌지만 지금은 4발 동물이 되어가는 단계 하나하나를 입증하는 화석들이 많습니다. 그 중에서도 결정적인 화석은 2006년에 발견된 틱타알릭입니다. 틱타알릭은 거의 완벽한 반어류, 반양서류 화석입니다. 틱타알릭의 비늘, 아래턱, 지느러미 줄기, 입 천장은 어류형 동물의 특징을 가졌고 짧아진 머리뼈 천정, 움직이는 목, 손목 관절 등은 4발 달린 육상동물의 특징을 가졌습니다. 귀는 땅 위와 물속에서 모두 소리를 들을 수 있었습니다. 틱타알릭의 발견 덕분에 지금은 물고기가 무투로 나오는 근사한 과도기 단계가 만들어졌습니다. 에우스테노프테론 같은 완벽한 수서성 육기 어류부터 양서류에 더 가까워진 형태를 거쳐 완전한 4발을 갖춘 형태로 부드럽게 이어집니다. 고래처럼 큰 동물이 마땅한 중간 단계 화석이 없을 땐 단골 메뉴에 오르기 딱 좋겠죠. 실제로 고래는 1980년대 초까지 고대 고래 화석들은 있었지만 고래와 비슷한 특징이 있는 포유류 화석이 거의 확보되지 않아서 창조과학 측의 집중 공격 대상이었습니다. 그러나 1983년에 파키케투스가 처음 발견된 뒤로 고래의 중간 단계 화석 발견이 빠르게 늘어났습니다. 파키케투스는 고대 고래와 비슷한 해부학적 특징들을 가졌는데 귀의 구조를 보면 물속에서도 소리를 들었을 것으로 추측되었습니다. 그리고 1994년에는 마침내 고래와 육상 포유류의 딱 중간에 있는 특성을 가진 암블로케투스가 발표되었습니다. 이후 달라니테스와 로도케투스, 타크라케투스, 가비오케투스 같은 중간 단계 화석들이 계속 발견되면서 유재류, 즉 발굽 달린 포유류에서 원시 고래까지의 전이 과정도 훌륭하게 설명되었습니다. 고래의 중간 단계 화석들이 갑자기 늘어난 것에 당황한 미국의 창조과학 측은 고래의 뒷다리를 물고 늘어졌습니다. 창조과학 강연자 주웨인 디시는 현생 고래에는 뒷다리가 없기 때문에 뒷다리가 있는 화석들은 고래가 아닌 다른 종으로 봐야 한다는 주장을 펼쳤습니다. 또다시 외형만 보고 화석 증거들을 부정한 것입니다. 하지만 기씨는 현생 고래에도 뒷다리가 있다는 사실을 몰랐습니다. 물론 기능을 하는 다리는 아니지만 볼기뼈와 넓적다리뼈의 흔적이 근육 속에 깊이 파묻혀 있어서 겉으로 보이지 않을 뿐입니다. 고래의 중간 단계 화석들은 숨어있는 뒷다리 외에 수백 가지의 해부학적 증거들로 입증되었습니다. 그리고 분자생물학적 분석으로 현재 고래의 육상동물 조상은 하마와 가장 가까운 것으로 알려졌습니다. 이 외에도 양막류에서 당국류와 원시 포유류로 넘어가는 전이 과정은 포유류의 폭발이라고 불릴 정도로 화석 기록이 풍부합니다. 종과 종 사이의 전이 과정을 연속적으로 보여주는 화석들, 특정 뼈의 진화 과정을 잘 보여주는 해부학적 중간 단계 화석들, 동물뿐 아니라 식물과 미화석의 중간 단계 화석들까지 중간 단계 화석들 이름만 열거해도 1시간이 넘는 영상이 나올 정도입니다. 중간 단계 화석은 과거에도 있었고 현재는 더 빠른 속도로 발견되고 있습니다. 고생물학자 도널드 프로세로는 현대의 화석 발굴 속도가 화석을 분석할 인력과 시간이 턱없이 모자랄 정도로 빠르다고 말합니다. 명백한 중간 단계 화석이 보고될 때마다 창조과학 측이 주로 대응하는 방법은 조작이라고 왜곡하는 것입니다. 그렇게 왜곡한 자료를 대중들에게 전파하면서 과학계에 대한 불신을 조장합니다. 화석은 발굴하는 일도 어렵지만 분석하고 검토하는 작업은 더 어렵습니다. 화석이 발견되면 수백 수천 점의 다른 화석들과 해부학적 비교를 하고 몇 년에 걸친 연구 과정 끝에 학계에 발표됩니다. 그 뒤로도 해당 분야 학자들의 혹독한 검증 과정을 거쳐야 학계의 인정을 받습니다. 이러한 검증 과정은 화석뿐만 아니라 과학계 전반에 자리 잡은 시스템입니다. 한두 사람의 조작으로 무너질 시스템도 아니고 수만 점의 화석이 가짜로 등록될 수 있는 시스템도 아닙니다. 과학 문명을 이끈 이런 시스템에 불신을 조장하는 단체가 있다면 참으로 슬픈 일이 아닐 수 없습니다. 책 화석은 말한다는 이외에도 수많은 중간 단계 화석들을 소개하고 있습니다. 해부학적 특징, 화석이 발견된 배경, 학계의 검증 과정도 재미있게 알려줍니다. 그래서 중간 단계가 없다고 주장하시는 분들에겐 필독서가, 나머지 분들에겐 재미있는 교양서가 되리라 생각합니다. 다시 한번 답글 대신 이 책을 추천드립니다. 이상 북툰이었습니다. 시청해주셔서 감사합니다.
Well, it's been already more than 3 years since this renowned scientist and great mind, Stephen Hawking, passed away. And he was quoted to have said sometime when he was alive as this, like, we are merely a little advanced from the waves. We can understand the universe. So that feature, that knowing that how the universe was made, makes us very special among, even if we were kind of the creature related with the apes and evolved from it. Okay, so we can actually elaborate this. So maybe am I just myself that's committing and creating fake news? Anything? So with the risk of such a behavior, I'd like to make a little bit of tweaks of what onto his original quote, like this. So instead of having this the understand the universe, also we can actually replace this. Not only understanding the universe, but also we know where we are from. Okay. And then that also can make us special. In more specifically, what do you mean by special? Make us very humble and compassionate. If we understand truly that where we are from, then it will definitely make us more humble and more compassionate to others. Like, rather, if we actually think ourselves as a really, really unique, some of the really blessed, single blessed identity, then we could feel like we could do anything onto others. But instead, we can take this kind of position, being humble and compassionate, which I believe, as a former, also myself as a really dedicated Christian, actually Catholic, to be more exact. I think this humble, being humble and compassionate are the two of the most important virtue of Christianity. Okay. Having said that, that's one side of the story. But also, what I think is, as I promised that I would say, make some statement about this conflict issue, about this religious belief and what the science have found within this boundary of science, of course. Like, look at this. This whole thing that we know as like science, in particular the natural science, only that's something that being played by the boundary set by the physics. Yes, that is really true. And within that specific limitation of what the physics has learned, this boundary, then the chemistry chemistry can move around only within this boundary. And with this physics and chemistry boundary that has been strictly set, then now it's time for this biology. That's what we are actually within. We are playing only this very limited, narrow territory of what we know. But out there, huge unknown, huge unknown out there, including, like, about actually speaking about the universe. Although present day modern physics, although they have made tremendous progress, but still there are so many things that this modern physics cannot even attempt to explain out there. So what I'm saying is, the whatever this evolution theory of evolution and or other rules, like including the rules of chemistry and physics, are all something that true and important, significant, but only within the small narrow boundary of this. Okay? But out there, here, so many other things. Maybe religion is here. Maybe also art, music, who knows, and philosophy. So, as a scientist, we should be very humble and we should be always careful. But at least, as far as we are playing in this, the territory of the scientific field, there are certain rules and the laws that cannot be violated. Something. That's all I can say about this. So, the last commentary about the fossil record is about the evolution of the horse, based upon the fossil record of the ancient common uh, ancestral form of a horse. Is interesting in that there was one kind of trend that you can find in overall. It is only overall trend that starting from the ancestor to the modern day horse, what you can find in as a general trend was the body size change to become bigger, and also the toe numbers has been reduced. So like actually this become a hoofed animal to have a fused toe, and also their size of and also shape of the tooth, in particular the molar, was the three major trend of this horse evolution. Here is in order to make a sharp contrasting comparison between the horse ancestor was named as a Hyracotherium and this modern day horse, scientifically is called the Equus, is what exactly as I said. So this was very small and also have the 4-digit toe as opposed to this one fused hoofed version of this toe, and the size of this teeth and their behavior, their habitat and their eating habit was also quite contrasting features. What's in it and the lying in between is actually the important thing is the climate change. So we can, based upon under the assumption of this big climate change that might have forced this transition according to what we know about the usual process of evolution. Actually, we could build one really nice story about how the evolution of this horse has arrived as it is in these days. So, but also, let me just go over this a little bit more in detail. So this Hyracotherium was their habitat was in the small forest, in forest as a small-bodied animal. So they were browsers, having soft, flesh leaves, and the small body size might have been beneficial to hide better from any potential predators. In those days, actually, this type of body shape and then morphological and anatomical feature might have been the best for them. But then let's imagine that somehow climate change entirely eliminated the forest. So these were all exposed to open field. Actually, there are some evidence that along with this the evolution, some evidence that several the big area of the forest that disappeared due to the drastic climate change. Then what? Then only those who had some of the genetic diversity who can quickly develop this quick transition. One transition is what? This to that. Why this? The 4-fingered toe would have been changed into one fused hoof toe. This would have been obviously like having one fused toe might have been more beneficial in running, because this, by trying to run hard, will very much easily you can actually have this sprained toe, but then you are actually like easy capture prey for the predators. So those who can develop this kind of feature, or some of them might have already this kind of a weird mutation already have when they were on the existence of this deep forest. But then those specific trait could play its effective role when whole environment changes. So then you can further refine and develop into the more emphasized such features. Same thing is also the body size, big body size. Yeah, like in the forest, when they were a forest dweller, smaller body size might have been much more like a plus. But then once they were exposed in an open field, the bigger the body is better for longer and faster running. So those would have been favored. And finally, what about this? change in this structure. Like when they were living in the forest, these soft foods were all available. But now, due to this quick change of this whole climate, now the only thing that you can eat is the grass on the field, which is much tougher to to be processed for the digestion. So obviously, if you have a luxury over this tougher is, then it might have been much more beneficial. So with that type of selective pressure from the environmental change, then now I'm not saying that some of this environment might have actually changed, then made some kind of live change, but probably what might have happened is such potential for those variation might be already present there even before those climate change, but then they were able to now enjoy some of the selection the selective benefits is probably how they evolved into this present form. But also something very important is, although we from like in the linear path, if you arrange all those different transition in a simply one linear way, then you can easily like be confused or like have a misconception, so to speak, misconception of, oh, they have some type of mission or directionality. So important thing that we shouldn't forget about is in the evolution there is no specific or intended direction in evolution. So if you thought that, oh, this hierarchical theorem was destined to become like this, starting from here to become like this, no, you're wrong, because as you can see in this diagram, there are so many different branches out there, like to have so many different ways of diversifications and were evidenced in the form of fossils. But for some reason, present days, the only surviving group of the modern day horses are somehow being represented by this particular group. It could have been also this group, but for some reason we don't have them, for example. So that's another like a casual and usual observation, and something that commonly we should take as a lesson from the evolutionary observation is that. And so those are some of these comparative morphology, anatomy, and with the help of this fossil records. But in modern days, the ultimate determinant of this assigning relative evolutionary relationship, that I could dare say that that this DNA sequence comparison is the thing. The most reliable way of building such phylogenetic tree. Why? Is because the difference actually, the DNA sequence difference among different individuals is highly correlated, proportional to. The more the sequence variation, the bigger the sequence the differences, then that means they are more distantly related. So here, even though this particular like animal, the classification, or you can say this as a phylogenetic tree diagram, you can build this phylogeny using conventional method like comparative or morphology and anatomy and fossil record, but this particular one was built entirely strictly based upon their relative DNA sequence differences among, right? And then it gave you the fairly accurate assessment. And so here at this point, with this occasion, you can do this phylogenetic tree building either through the conventional way or through this DNA sequence comparison. However, especially when you try to do this same thing in, like in the case of having such a similar thing in among different bacteria, like this, where you cannot really have such the clear clue about their comparative morphology or anatomy like stuff. Then the only alternative, correct alternative, is a DNA sequence comparison. So you better actually have some kind of a strong theoretical foundation for why you can legitimately and reliably utilize this DNA sequence comparison in comparing their evolutionary relatedness. What we can say is, as I already put it, if the DNA sequence difference is greater, that means they are more distantly related. Why? Why is that so? Because it's totally based upon the one thing, the logically speaking, is based upon the mutation. Yeah, mutation is the one actually providing this DNA sequence differences. And mutation occurs randomly, and also it is something that has a constant rate, so it means it is a kind of function of time. In other words, let's say it's like I always use this comparison of identical twin. The case is this: let's say there was an identical twin was born initially, then it means at this stage, if you compare the DNA sequence of these two identical twins, then would be, I guarantee it will be 100% identical at this point. However, if this identical twin lived something like 100 years after, when they have this reunion in celebration of this 100 years of this age, one might suggest that how about like we have compare DNA sequence comparison. Do you really think that once again after 100 years their DNA sequence will match 100%? It may not at this point, because individual A and B, even though they are identical twins, so at birth their DNA sequence was 100% identical, but over those years of living might have created the differences in DNA sequences through mutation. And if this longevity continues something like 5,000 years, for example, wow, I wish I could do that, live up to 5,000 years. As this angle will would be widened up, like that will be an indicative of this DNA sequence differences potentially. So after the 5,000 years, if they were still alive, then probably, definitely, most definitely, their DNA sequence will be quite different, so to speak. And this is like a perfect analogy of what's happening from this common ancestor. So that's why the DNA sequence difference can be used as a direct indication of the time since these two group of organisms has a split from the previous common ancestor form. Is the rationale or the theoretical rationale of this. Okay. Yeah. So finally, although I know it's been already been a while, and then this is a third section of the lecture, but this is the last section of the this chapter, which is going to be about the origin of human evolution. So let me finish this part. So if you look at this chimpanzee and human, yeah, they are. We are told that chimpanzee and humans are the closest relative of the two animals existing on Earth right now. So let's take a look at our history of evolution to become this, and then separate from whatever those our close relatives. We belong to a relatively bigger group of primate, which the primate, this group has a small group of the mammals, especially these eutherian mammals, like placental mammals, and this group includes the lemurs and tarsiers and monkeys and apes. I didn't know actually what lemur was, and then today I actually found about this. Oh, lemur was this. Actually, my best friend's nickname was this lemur when in those high school days, and now I understood. And although I really miss him because he just passed away at the age of 35 years of old due to this liver cancer. But anyway, I really miss him. And this tarsier, it's another monkey, and so like this. Also another smaller group we can among all these primates, these particular apes are the smaller group to which this modern humans also belong, and also include this our closer relatives, including chimpanzee and gorillas and all other type of monkeys too. So it's been estimated that we've been split from chimpanzee about this type of this kind of period of time, like 4 to 6 million years ago, and then established another independent group now called the hominins. So in this hominin, that includes humans and other all those extinct relatives. So we are the only member of this hominin presently still ever alive. So those include one of such famous hominins include the famous Australopithecus and Homo habilis and Homo erectus, Neanderthals and Homo sapiens. But these are not the only those hominins, these important things. So there also have been some of this evolutionary trend we can notice. Right? Another thing is, once again, if you please, we should not actually try to view in this way of a linear way of evolution from starting from this Australopithecus to becoming this modern human Homo sapiens, but we have to be aware of this feature of always there has been some of the particular hypothetical common ancestor were there to make these two relatively related forms be split from it. So this bigger trend of this transition or evolution was this. One is a behavioral or the physical feature of that we can summarize as a bipedalism, like standing upright and walking with only two legs is a bipedalism, and another is an increase in the head cranial size, or kind of can be recognized as the important trend. And all of this, our modern humans or living humans, are believed to be derived from the Africa, originated from Africa. According to this theory, initially the earlier form of the human, the ancestor form of human, Homo erectus, migrated out of the Africa to Europe and Asia, but they failed. But only leaving the evolution of this Neanderthals. So these are actually rather kind of a direct descendant of this original Homo erectus, came from Africa. But then there are second event of such migration more recently, so like about 150 to 200,000 years ago. Another group of modern humans also has migrated from Africa, and this time this timeline represent the another such event of migration. So although the initial Homo erectus migration from Africa has failed to make any kind of evolution, continuous evolution, but the Neanderthals. But this time, all these descendants of Homo sapiens could settle in all other places of the rest of the place of the earth to become the foundation of this modern human races. So, in other words, already, especially at least in the Europe and some Asia form, that there might have been some existing Neanderthals there. So, for a certain significant period of time, this new modern human must have coexisted with the existing old the residents, that is Neanderthals, especially in Europe. So this is the map showing such a migration originally from this route. So what you can see over here is starting from this, not only having this out of Africa migration, but also evidence that show that this is approximately the original epicenter of that such a human origin, and then also spread in the Africa. So not only this earlier older form of these humans, including Australopithecus and some of the more later form of the humans like Homo habilis and Homo erectus. By the way, this Homo habilis means like they are the one who first ever used these tools, hand tools, and Homo erectus. Then you can probably infer that these are the one who first more stably, more completely have this upright by practice the bipedalism. And then even the more recent Homo sapiens, including these Neanderthals and Homo sapiens, sometimes actually this Homo neanderthalensis is also called another, usually some other occasions, they are also called Homo sapiens and neanderthalensis as the subspecies name, neanderthalensis. Homo sapiens neanderthalensis. And on the other hand, this us is called Homo sapiens. Sapiens is the way they are distinguished and called. But here in this particular occasion, they are called Homo neanderthalensis. Because the problem over here is, by just looking at this Homo neanderthalensis or Homo heidelbergensis or denisovans, all these indicate they are completely separate different species. But according to the biological species definition, especially when they have this coexistence period, they were able to have this interbreeding. There was no problem. There was no anything kind of a barrier preventing them from having this kind of interbreeding. So we'll get to that later. So this type of how and how what are their relative relatedness, and also how these humans migrate from one place to the others, all these things can be traced by DNA sequence comparison, of course. But then in particular, the comparison of DNA sequence in particular is in the mitochondrial DNA and also Y chromosomal DNAs. These are the two very useful, instrumental in deciphering those the relatedness. So, in particular, their origin, modern humans' origin, and their relative relationship. Also, even today's, like racial relationship. For example, in Korean, as Koreans, are we more related to Mongolian, or are we more related to the Japanese type of thing can be done by this DNA sequence comparison, in particular mitochondria as well as the Y chromosome DNA. Why? The benefit of using mitochondrial DNA is once, first of all, as I have already mentioned, that mitochondria contain their own independent genome, the DNA, like this. So as you can see, if you zoom in a cell, you have several copies of, actually many, several, even several hundred different copies of a mitochondria can exist in a single cell. And within each, you can have multiple copies of this their own circular DNA, their own genome. It's their future, and that's one advantage of using mitochondrial DNA in this particular purpose. And also another advantage is this: they don't have any things like this recombination. What is recombination? This recombination is something that only the DNA in the nucleus of a new eukaryotic cell. Only nuclear DNA can have this recombination event during the gamete production. What did I say about the gamete production? It's called meiosis, isn't it? The cell division leading to the gamete formation is meiosis. And during meiosis, something called this homologous chromosome. These homologous chromosomes are the same chromosome but having the different origin. This chromosome, let's say if this chromosome came from your father, paternal origin, and the same chromosome, identical chromosome, must have come from the mother's origin. Okay, so actual more details, the characteristic of this homologous chromosome, we will get to that later in different chapters. However, here at least recognize that there are certain things called the homologous chromosome. Why? As this individual must have been diploid, right? As a diploid individual, you have two origin of this your chromosome set. One is from father and one is from mother. So this identical chromosome can physically align. So this alignment occur during meiosis. Okay, not in another type of cell division, mitosis. We will get to that later. So during this gamete formation, when they are aligned like that, and they do have this type of event called crossing over. And after the crossing over, when it was resolved in this way, then they have this exchange of their segments. Although basically they are same chromosome, but details nature of these two chromosomes can be different, of course. Why? Think about it. Because these two chromosomes' origins are different. Your father and your mother have lived independently from their lineage for a long time. So how can you expect that actual DNA sequence between these two homologous chromosomes are 100% identical? Not necessarily so. So in this way, you can have this kind of a shuffling of DNA sequence. This is what you call this recombination. So here, this recombination event between two homologous chromosomes are described. in this figure, which I just drew myself. So because of that, nuclear chromosomes will have this recombination. Not that is something that a bad thing, which is actually a good thing for increasing the genetic diversity in the offspring, which is recombination is very good. However, in this particular purpose of comparison the DNA sequence among different individuals and then to determine their relative relatedness in evolution, this recombination will have a bad influence because you have to assume actually there isn't any recombination, and then only mathematically you can predict like based upon the DNA sequence difference, and then recombination will have a like add-on effect, make this assessment a little bit inaccurate. Okay, it's not something that like a game changer. So still with the recombination you can have a pretty much a fairly accurate estimation, but still ideally speaking. But in mitochondria, a good thing is you don't have any such thing like a recombination because the mitochondria is not a diploidal in genome, so it is a haploid genome. I will explain why mitochondria is a haploid genome, unlike the nuclear genome. It is a haploid. They don't have mitochondrial DNA does not have any homologous chromosome, right? So they don't have any homologous chromosome, so it is a haploid, and they don't have any recombination. So any sequence variation is absolutely you can assume is coming from the mutation only. So it is a very accurate estimate of the differences, right? And another advantage is mitochondria. As you can see, in a single mitochondria you can have multiple copies of this DNA, and also in a single cell you have hundreds of different multiple mitochondrial copy. So especially it's become much beneficial to try to have this DNA sequence examination from really hard samples like fossil. Yeah, you can expect that you won't be able to get too much DNA sample extracted from those dead fossil samples, but still mitochondrial DNA will enable this. And another thing is this mitochondrial DNA is, as I have mentioned, is being inherited only from mother to the sons, and so although whether you are so the origin is always from the female origin, so you can trace, especially trace this origin, the lineage of the maternal effect can be easily traced. The reason why you only get your mitochondrial DNA inherited from your mom is because of during this fertilization, the egg cytoplasm only get to contribute to the zygote. So anyway, so it is very ideal item to trace the maternal female origin lineage. So actually this is a lot of times is called the mitochondrial Eve project. So like to trace back to the origin of this actual Eve in the Bible, you can easily use this mitochondrial DNA comparison. On the other hand, the human Y chromosome is something the opposite. It also has no recombination, and in this case it gets to be transmitted only from male to male, because obviously human male have this Y chromosome only, females have two X chromosome. That's the reason why we can say this Y chromosome does not have any the recombination event. It's because Y and X chromosomes are totally two different chromosomes. They don't have any sequence similarity, virtually, practically speaking. So only those recombination can take place between two same like chromosomes, like two X chromosome here. The recombination can occur. But here, between Y and X chromosome, recombination cannot take place. In the case of a human, like frogs, they can, because frogs' Y chromosome is different, so that it is kind of homologous to the X chromosome. But in high mammals, X and Y chromosome, they are not actually homologous, but they still align together during this gamete production. So once again, anyway, and the result is human Y chromosome can also have no recombination, so which is very good for this purpose. And it also their transmissions only from male to male, so it is convenient to have the trace of the paternal origin. So a lot of times it is being referred as so-called like Y chromosome Adam project. So speaking of that, using that, actually the Neanderthals and the humans genomes have been compared. Actually, before this so-called Neanderthal genome project, which was occurred in 2006, like 4 years before, already the human genome was entirely deciphered through the Human Genome Project. So we already know the exact DNA sequence of human Homo sapiens. But then 4 years later, this very challenging project has been launched in Germany, in Max Planck Institute. So this DNA, the Neanderthal genome, also consists of this like 3 billion base pair, the nucleotide, 3 billion nucleotide base pairs, because they are DNA double helix DNA sequence. So identical to 3 billion in numbers, identical to that of humans. Okay. It's been sequenced, okay. And it took only 4 years. Like for this, the same comparison, to do so, the same thing, even though it must have been much easier to obtain the DNA sample in the case of human, it took more than 10 years. Actually, in reality, it took 13 years. But here, with the progress of the technology, it took only 4 years getting the sample out of those. Only available sample, DNA sample you could get from Neanderthal is a fossils, but still they were able to do that. And in this case, even they're not something that used the mitochondrial DNA, the entire genome DNA. Mitochondrial DNA is very small in size, but entire genome DNA is huge, like 3 billion DNA base pair, as you can see. But then it turned out that, wow, it showed 99.7% percent identical with human. In comparison, in the case of a chimpanzee, it shows a 98.8%. So even in the case of chimpanzee, was a striking similarity between human and chimpanzee. But then here, like Neanderthal, is like no wonder. So if they were still alive, yeah, it is like de facto the closest relative, like brothers, to modern humans. But the more important thing is this: interbreeding. It's been revealed that once you complete this DNA sequencing, then you know that how much you can actually determine the sum of the area that you have in human genome actually came originally from who? The Neanderthals are the thing that we have learned. Okay. So, but that's not it. Not only the Neanderthals, but some other modern humans, including something the group of these modern human, they are all extinct, but Denisovans and also Homo floresiensis, its name is nicknamed Hobbit because it turns out that they have such a very small statues, and also Homo Heidelberg Heidelbergensis. How can I just not being able to pronounce properly? Heidelbergensis, right? Heidelbergensis, whatever. All this actually showed significant degree of this gene exchange, which means they have interbred. They produce the offspring between across this barrier boundary of this their species, okay, especially more extensively with this Neanderthals. So you can actually have this kind of a map. So here, like towards this end, the red end is having the larger proportion of this Neanderthal legacy of the genome, and towards the blue area, Denisovan's legacy. So on this area of a map, you can find so many such the remnant traces of such gene interbreeding. One interesting thing is here in Africa, you don't have any such thing, right? Actually, the genetic diversity, if you compare the actual genetic diversity, those who currently living in Africa, the native Africans, are the one who actually who shows the most diverse in genetics, in their gene diversity, in DNA sequence diversity. That means those who now living in the outside of Africa are the one actually originated from very small group of initial the ancestral form came from this Africa is the message that we can take. And so, and one particular characteristic about this Neanderthal genome legacy left on us is a lot of those genes are the bad genes, so-called, like the genes that causing a depression. Wow, genes responsible for causing the allergy response, and this circulatory defect, like including heart attack and this diabetes and addictions. So many are all turned out to be originated from Neanderthals. What is this? Why did our ancestors have such a mistake? Such a stupid mistake to have such a interbreeding, so that we have this legacy of suffering. So, in other words, does that mean Neanderthals? So, like all those bad genes are like we can just point fingers to the Neanderthals. Does that mean that they were such an inferior race? So that's the reason. No wonder why they got extinct. Is that kind of a proper way of the interpretation of this? No, I don't think so. Is that means simply by the time they leave the Neanderthals, the habitat environment, these genes, like in present environment, these genes might have actually caused some of the troubles, but back then those genes were not at all actually something that troublesome, but actually on the contrary, even some of those genes might have been actually helped for them to survive through such harsh environment. Is probably what we have to get as a takeaway of this. So with that, with my sincere apology, I can end this week's lecture series. So the only excuse is this, although I have a lengthier lecture for this week, but a lot of time, a lot of those portion were taken by the movie films, inserted the clips. So that's kind of a little bit of a kind of a excuse, please. So then, please, don't be angry, and so just still have a nice weekend. Bye bye.
Can I go back to the days that I had you? back to one more not like we used to. I miss every minute we've been through, from the hardest part to the best you. Make the stars align, words cannot describe. Don't need no more goodbye.